Good morning. Welcome to the Boardwalk Talks series brought to you by the aquarium at the Dauphin Island Sea Lab. My name is Mendel Graber. I am one of the educators here at the lab, and this morning we are going to be chatting with Dr. Ryan Moody about seashells, about mollusks, about molluscan diversity, about collecting seashells, and about a shell collection that is housed here at the Sea Lab. And some of the challenges that we have faced recently in recovering this collection after we had some roof damage from Hurricane Sally last fall. So with that, I am going to let Ryan introduce you to the world of mollusks.
Yeah, so good morning. I thought what we'd start with is just talking about what makes a mollusk a mollusk. So they are animals without backbone, so they're invertebrates. And despite being extraordinarily diverse, they do all have a few things in common. If you picked up a shell at the beach, one of the most obvious, of course, is the fact that they have a shell. Mollusks have a shell, of course. In some of the groups, it's reduced. If you think of something like a squid or an octopus, a lot of those do have remnants of a shell, but they've traded the armor of a protective external shell for speed and agility. They have a special tissue called mantle, which actually secretes the shell, much in the same way that we have cells that secrete our skeleton. They have a specialized feeding structure called a radula. A lot of people are familiar with the fact that sharks will constantly grow teeth, and if a tooth breaks off, it regrows. Think of a radula as a strip of sandpaper that just continuously grows, and if you've ever seen a snail feeding on the side of an aquarium, you'll sometimes see this little scraping structure. That's its radula. And again, even the octopus and squid, they have them within their beaks. And then, last of all, they have a muscular foot. And if you've seen snails crawling along in maybe in your backyard, it is just that flat surface that they're secreting mucus from and moving along. In squids and octopus, the arms and tentacles that allow them to manipulate things and move about, those are derived from the same structure. That is their foot, except it's a series of arms and tentacles. So those are characteristics that make mollusks mollusks. So within the phylum Mollusca, it is our second most diverse phylum of animals. What these animals do? Well, first off, you can find gastropods, bivalves, cephalopods worldwide. They've done very well. You can find the snails on land, on the coast. You can go collect snails at the beach, the deep sea, from the tropics all the way to the poles, Antarctica, the Arctic. Bivalves, they have to be in water. You're not going to find a land bivalve, but again, you'll find those everywhere from coastal areas to the deep sea, from the tropics all the way to the poles.
And in freshwater.
The bivalves, yes, are also freshwater, and as are snails. We have quite a diverse assemblage of freshwater snails as well. So the point is that mollusks really have spread just about everywhere. They have adaptations to live on land, just speaking as a group, land, freshwater, marine environments. Whether it's cold, it's hot, they've diversified, and we use them for food, for decoration. We have lots of commercial uses for them. But as far as what they do, most bivalves are, so if we think of something like a scallop, they are filter feeders. They are eating small single-celled organisms from the water. Snails do a huge number of things. There are herbivorous snails, carnivorous snails. There's some filter-feeding snails. The snails that live in trees, and they're feeding on various materials up there. So they've taken advantage of a huge number of habitats, a huge number of diets that they're able to use, and that's part of the reason that they're so successful as a group.
What's the longest dated back snail that's been found, or mollusk? If you were
like in the fossil record?
In the fossil record? So the relatives there seem to show up in the Cambrian, so around 550 million years ago. That period we refer to as the Cambrian Explosion, because all of a sudden you seem to have a lot of organisms in the fossil record. And it's not really that they weren't there. That's the point in time where you see the evolution of hard parts, shells and skeletal structures, and those are what preserve really well. And before that, you just end up with these little imprints of soft-bodied animals. So we do have fossils of life before that, but it's in the Cambrian that we begin to see these very hard structures, skeletal components and shells that fossilize really well.
So what is this collection that we have here in front of us?
So
hang on, before we move on
on
to the collection, let's just talk a little bit about, you talked about the diversity of habitats that they occupy. Can we talk a little bit about the diversity of form that is related to this diversity of habitat?
So if we take snails, let me get my wire here. Let me see. I thought we had this open. We'll take a look. Let's look at some of the a little older groups. We have things like abalones, which on top of being delicious are really pretty. This is that iridescent mother of pearl material called nacre. A lot of these animals, you can have all sorts of little ribs and bumps of different types, but they're relatively, they're going to keep the same shape. They're not super ornamental. They have holes that help direct water flow for respiration. In some of these holes, water's coming in, passing over the gills, and then passing back out. We see, let's see, we have this guy bagged up. Here's a slit shell that uses a slit in the shell to help direct water up over the snail's head would come out right here, and water will pass over its gills, and then waste gets carried away right here at the end of this slit. And we then start to see snails that are relatively thick-shelled, but again, not a lot of those, that ornamentation on the surface. And it's because if you look on the inside with this nacre, these snails can't really resorb or dissolve and reuse any of the shell material on the inside. So you have, somewhat simplistic shapes to these snails. Compare that, even though this is really pretty, there's some bumps and grooves, compare that to a snail like this, which does not have that mother of pearl nacre, the iridescent layer, and it's able to remodel its shell and redeposit shell material as these wonderful spines and ribs. And there's been a lot of work into why these snails have the types of shell sculpture that they have. And if we look over here, we can see these can get pretty crazy in terms of spines. In most cases, these spines serve a protective function. If you look at the size of the snail, he's about this wide, but if you've got spines, a large fish such as a ray, crabs, it's one thing to crush the snail when it's trying to crush it here this wide. Well, now suddenly to the crab, it's going to have to reach around the entire spine. It becomes far more difficult. So there's protective structures. Go ahead.
Let's go back to basics for just a minute and point out that snails make their shells.
Yes, so that's another important thing. It's a common misconception that when you go to the beach, you often will find snail shells with hermit crabs in them. The hermit crabs did not make the shell. The snails, they secrete the shell. And Mendel has some baby lightning whelks here. This is their skeleton. You can't remove the snail from its skeleton. The muscle, there's a muscle that attaches it to its shell. And it is when the snail dies that other animals can make use of the shell for their home. And so just really quickly, she's got a wonderful cross-section shell. If we could just zoom in here. The way that these animals are growing, and it works the same way with bivalves, excuse me, the animal is going to, with that mantle, remember the mantle tissue is the tissue that actually secretes the shell. And what it's doing is adding new shell just right here on this edge. And it will grow around, and you can see what the snail looked like on the interior here when it was young. So if you matched this up, you can see that this little whorl right here of spines was probably, that's what the snail would have looked like. And if you flip it, you can even see that this portion of the shell, when it's young, becomes an interior portion when the shell continues to wrap around and around. So the snail is attached quite a ways in by a muscle, and if you pull the snail off of its shell, it's going to die.
So this is a lightning whelk egg case.
Yeah, here are the egg cases. They will usually have around a dozen, well, usually around 100 eggs in them. Only about 10 or 12 babies will emerge from each of these little capsules here. Are these full? I think they're emptied out. But this is what will come out. These are tiny little baby snails, and again, they just grow their shell around and around and around, and that's how they're going to grow until they're quite large. If you look at a bivalve, bivalves are doing the same thing. If you look way up here, you can see what the bivalve looked like when it was young. And so here, the bivalve with its mantle tissue is adding its new shell in layers right on the outside so it grows outward.
So then you were talking about the way different snails and also clams grow, so they can have pretty dramatically different. So these are different kinds of snails, and so the form kind of is related to the lifestyle of the animals.
Yeah. So snails, so first off, they can live anywhere from soft mud, sand, rock. And if you've ever found, this is a cowrie, and if you've ever seen snails that are very, very smooth and glossy like this, the mantle actually comes up and wraps around the snail. And that's what you're seeing, the tissue, the soft parts of the snail. In these guys, the soft parts are always going to stay within the snail, or they'll crawl around like a garden snail that you might see in your backyard.
So another thing, so you were making note of the research that has been done on the reason for this diversity, and we would make the logical assumption that this is a good defensive structure, and that has been supported through research
for different groups. But let's look at this guy. This is a snail that lives in very, very soft mud, and despite being very spiny, those are pretty fragile spines, and it's thought that these kind of act like snowshoes on very, very soft sediment. So when the snail comes out, the body's also kind of protected here in a cage-like structure under those spines. Yeah. And this gives more surface area, so it doesn't just sink down into very, very soft mud. So lots of reasons. Yeah, this is a giant ton snail, and you can find you usually find pieces of these on the beach around here from time to time. Very thin. Yeah.
So just to
can we hear the? Oh, you've got microphone.
So I'm doing that very gently because I don't want to crack this shell, but you can kind of get an impression of how thin this is compared to, with this one I can. Like if you can hear it, this shell material is much, much more dense.
Yeah. You can see just how thick they are.
If you're looking at this, the primary function of this shell as protection, you might wonder, why don't they all have a really thick shell? Why do some of them have a really thin shell? So can you comment on why mollusks do not all have a thick shell? Why do some of them have a thin shell?
Part of it is, here, let's look at a bivalve example as well. This is a razor clam. They dig very deep. There's some.
Look, I have a pickled razor clam.
Oh, she's got a pickled one. So some clams sit on the surface. Some will burrow quite deep, and you don't really need a very thick shell. Why? It takes energy to make a large, thick shell. You also have to carry around that large, heavy shell as well. So you can see that the ton shell here, for example, that animal it's going to fill that shell. It's going to be quite large, but it has a much lighter shell to carry around, and he's going to be partially buried in the sediment, and
then the
count on that to
the
avoid
ridges
predators.
kind of split the difference.
Yeah, and the ridges make it quite strong for its mass. Yeah.
And for it's a lower energy expense, but it still kind of gives it a little more protection than one without ridges.
And then, of course, we can get really weird. Here we have this is in fact a bivalve. This is find these in deeper water. So it is a bivalve, but its second shell forms this little perforated cap. That's the other valve. This is called a watering, what's the common name? They're called Burchides, or a watering spout or watering can bivalve. And they'll usually live in the sediment and sit just like that. And they're filter feeders. They're in the deep sea. There's not much that, there's not as many crabs and other animals that are going to come along and pull it up and eat it.
Is there anything that comes out the top of it?
That's just where water passes through.
So, Ryan, let's review this for a second, because there might be people who wonder, what makes this a bivalve? It does not seem to follow the bivalve body form. As taxonomists would identify certain features that would make a bivalve and not a gastropod.
Yeah, well,
and so to look at this, this doesn't look like a bivalve. So what features make this a bivalve rather than something else?
And that's why we go back to what we started with.
Mm-hmm.
They have shell. They have mantle tissue that produces that shell. They are going to have gill structure similar to other bivalves. There are lots of anatomical similarities.
Mm-hmm.
And of course we can do DNA as well.
And then
but as their larvae, when they're larvae, when they're tiny little babies, they have far more, if I recall, many of the same characteristics.
And we should also mention that sometimes taxonomists rearrange their organization systems
Constantly. The scientific names for a lot of the mollusks change constantly. Yes.
And then I think you should
show
this one too.
So this is a spondylid oyster or a thorny oyster. Lots and lots of spines. These guys, just like our oysters that you would find in reefs that form on the coast here, they will concrete themselves. They'll essentially glue themselves with shell to other oysters or to rock. These guys do the same thing. These are close relatives of our oysters, but they're spiny. But if you found this guy while you were snorkeling or diving, he's not going to look that pretty. He's going to be covered in algal growth, sponges, and it's very, very difficult to even see it. So there's camouflage that goes into this as well. You have all of these spines where other organisms can grow. Oh yeah, and so we wanted to mention also, sometimes if you find very fresh shells, both bivalves and snails, you'll sometimes find this kind of furry layer. It's a protein, it's flaking away. It's a protein layer called the periostracum, and it helps with camouflage. It helps, things can grow on that as well to increase the amount of camouflage, and it also helps prevent other organisms like small sponges and worms from burrowing into their shell. So it's got protective functions both from things settling, things like barnacles. Barnacles like to grow on everything. Do you have an oyster with barnacles by chance?
Well, this one has some worm tracks.
Here's some
and
worm tracks
in it. I might have one with barnacles.
And on these bivalves and snails with that very thick protein layer, the periostracum, that's really fuzzy in some species, it helps prevent worms from burrowing down or barnacles from settling and growing. So that would be, having a shell full of barnacles would be a lot of extra weight to carry around.
So you've brought up some of the diversity of habitats that mollusks as a group inhabit, but just to re-emphasize that we have just touched the surface of the
this is another really weird one,
diversity of habitats.
And as odd as this guy looks, these burrows are made by another bivalve called a shipworm. And if you see these live, you would be forgiven to mistake them for worms because they have big, long, soft bodies. But up at the top, they've got these two valves that interconnect, and they move back and forth like a drill bit and work their way through the wood. So their two shells are just at the very top. and they've got little serrations on them, and they'll burrow and make their way.
So this is not a shipworm. A shipworm is a clam. This is a different kind of clam. This is a razor clam. But just to give you an idea of that muscular foot right there.
So that's what the foot looks like. That same structure that we were saying the snail uses to move along, that's the same structure in a bivalve.
In the shipworms, the foot is proportionally longer compared to the shell. So the shell is smaller, the foot is longer. So they resemble worms, which is how they got their common name, but they are not worms.
And this is also a good one since it's out. You can also see in the bivalves they have siphons. Water comes in. One of those siphons passes over the gills. For both respiration, we need to get oxygen and get rid of carbon dioxide, and to collect food. And then water goes back out the other siphon. Species that dig really deep in the sediment, usually in sand and mud, will have much larger, more well-developed feet and much longer siphons.
It's kind of like a hose. The siphon is.
Yeah, and that passes down over the gills.
So then you want to tell us a little bit about this collection?
Yeah, so this collection was donated in the 90s by a physician, Dr. Warren, from lives across the bay. And what is impressive about it, number one, the sheer number of specimens, but that there are representatives of bivalves and snails. We talked a little bit about the nautilus. We've got representatives from all the different major groups of mollusks, but also from all over the world, from the United States to all around the Pacific Ocean, the Eastern Atlantic, and Africa. Yeah, this is another group of mollusks. This is just the shells that they produce. This is called a chitin. And in life, there is tissue that would wrap around and hold all of these shells together. They always have 1, 2, 3, 4, 5, 6, 7, 8. They always have 8. And kind of move around like in a slug-like fashion. But this is another class, just like we're looking at these larger groups like the snails, the bivalves, the cephalopods, like that include the squid, octopus, cuttlefish. The chitins are yet another big group of mollusks. This is a fragmented piece of what's called a paper nautilus. This is a species of octopus that will produce this really beautiful shell and lay its eggs within that.
So the incredible diversity in this collection of the different groups, and then also representatives from around the world.
And it can get overwhelming. I mean, we're just trying to scratch at the surface and show some of the major adaptations you see in these groups.
What is the importance of a collection this large? You as a mollusk person, when you look at something like this, there has to be a part of you that, like, it opens a door to you wanting to know more about something.
It does. You get to see organisms from all over the world that live in all of these different habitats, and you can start to see these trends in shell form that reflects their adaptations to where they live and what they do. And one of the things we wanted to show over here, one of the most valuable parts of this collection is the fact that, let's point, before I show you this, we do need to point out that everyone always likes to know what is the largest of anything. And here is this is an Australian trumpet. This is the largest species of gastropod in the world. So there you go. And this is what it looks like when it was younger. This is a baby. And again, you can match that up, and if you cut this shell in half, you'd be able to see that shell form when it was smaller.
How long can that live?
I am not sure how old these get. These are probably on the order of well over a decade. I'm not going to go beyond that, but I would say probably 15 plus years, if not older. There are clams that can live for a very long time that we find in cold water environments. That's a tropical environment. But this unassuming box is actually one of the most interesting things about this collection, because if we look in here, all of these specimens in this collection are numbered. And if we just pull a random card, we can see the species, we can see the range where you would find this particular species, but where this particular specimen, so this card corresponds to a particular shell in the collection. And there's information, 10 feet. This was collected at 10 feet in Puerto Rico. The date, so in September 1983, all of this information is available for every specimen in this collection. Where was it collected? What was the habitat? What was the depth? Some of these were purchased from around the world, but the data is there as well.
Can you give a specific example of what scientific question might be answered with data like this?
Well, you might ask, there are some species, for example, that you only find in very small geographic areas. In a collection like this, where these dates go back into the 80s, probably a little earlier, he might have specimens from a particular location of a species in the Pacific, and there could be observations that this particular species hasn't been found in some time, or maybe they thought that it had disappeared long before one was collected and put into this collection. But so we can find out where organisms were distributed in time and have a record of that.
So someone who is knowledgeable about the scientific body of knowledge of mollusks might be looking through a collection such as this and, for example, be surprised to find that that particular mollusk was collected in that particular place or at that particular time. And it might be kind of a discovery, this is unknown in the scientific community that these specimens that this species is found here.
Yeah.
So, I mean, for example, you might have some fishing communities that maybe have collected specimens like this, but it has not been made part of the scientific record just because.
Yeah, and that happens with a lot of organisms. The coelacanth, the big lobed fin fishes that you find off of Africa and Madagascar, where they found more on the eastern side of the Indian Ocean. They thought they were extinct. They were only known from the fossil record. And then all of a sudden, some ichthyologists, some fish biologists went to a seafood market and said, I know this fish, but only from the fossil record. And they realized, oh my gosh, this thing's still alive.
So the specimens themselves are really fascinating, but the data that goes with them is really valuable.
Yeah. And a lot of people love to collect shells. They love to collect shells, or are interested in plants or fungi or birds. And just taking that extra step to record where you saw it, the habitat, the date, that is extraordinarily valuable. Even if you're if you're a photographer, photographing birds, just make a note of where.
We have a question from Jason Logue. He asks, will this data be input electronically and made available for other scientists?
There are a lot of people that do produce these big collections, and they do this for themselves. This one, I think we are going to end up, and you can speak to that.
We'll come back to that.
We'll come back to that. Yeah, this one I think is going to get electronically inputted. I don't know about putting it on the web and making it searchable, but ideally that would be the best thing to do.
How many shell collections like this do you think are in the world?
There are lots of hobbyists. Yeah, so there are scientific, actual scientific collections where species were collected from a certain region for various research, to address certain research questions. There are other people who just travel the world and collect shells and put these together, and oftentimes they end up just staying in their house or might be known to a small number of people.
So then let's bring up a resource like iNaturalist.
Yeah. So iNaturalist is great because all of those things we just said, where was it found, when was it found, all of those pieces of information, not only...
Well, let's start by telling people what iNaturalist is.
Okay. Yeah. Well, I was going to say that that's usually done primarily by photographs. So, I don't know, you take a picture of a plant. It's an app and a website, and you can make create an account where you can go out and I think it treats everything from plants and animals and fungi, you name it. It even has a search function to help identify an organism. But using a photograph, and most smartphones now will go ahead and include the
location data if you allow it,
the time, all of those things. You don't even have to think about it. You can upload these photos, identify them, and other people in the community will help you identify, verify the identity of a particular species, and it's now on the internet. I mean, it is a searchable, accessible database. Yeah, whatever I'm trying to say. Yeah, it's a database that anyone can contribute to, but also
access, search. It's pretty remarkable.
Yeah, you can say, hey, I found this insect. You might realize, oh no, this is just found in Southeast Asia. This can't be the one, right? I mean, and you get to see a map where all the different collections have been made for particular species. So that's kind of the most modern way of dealing with this sort of information.
Collecting the data.
Yeah, collecting it, putting it into a database that anyone can not only contribute to, but also access.
Yeah.
So a question in some of these shells, you know, if it's found in 1983, is there a way to do some type of isotope look to it to maybe find out more about the waters that it was in?
There might be. It would require destroying part of the shell to do that, and that would probably only be done if there was some very specific question that was being asked for a particular species. Yeah.
So to come back to this collection, again, it was the building that this collection was in had roof damage from Hurricane Sally. And so there was water infiltration into this building, and the collection was impacted by that. So one of the, the urgent things to take care of in recovering and securing this collection is preserving the data, which is, on these paper labels. So they are very susceptible to mold and to insect damage and to sunlight fading them.
If you want to take a look, this is an example of a tray where you can see the species name, the location where it was collected, but if you notice there's also a number associated with each of these, and those numbers correspond to a card in the catalog with additional information, and that's the most valuable part of this collection.
So Randy, do you want to come over and tell us? This is Randy Cannon.
Of course she does.
And Randy is a grad student here, and Randy, I don't think you are doing your graduate work on mollusks, is that right?
I'm definitely not.
So she has been working really hard to recover this collection, but it is a monumental undertaking. So let me just quickly show a picture of what they have already, and you can talk about the process for what you guys have worked on here.
So we have been working essentially drawer by drawer, and I will remove a drawer, clean the shells, and then, like Mendel and Ryan have been saying, the most important part is the labels. So I rewrite the labels that go with each of the shells, and then we restore those shells in either a box or a baggie, whatever fits them. So if it's more of a sensitive shell, I'll put it in a box. If it's more of like a stronger shell, we can put it in a bag, and then we put all of those in new boxes that are labeled to match the drawer that they came from. And so yeah, that is the most recent bin that I just completed. It's bin 41. So we now have 41 of these bins that are saved.
So you as a graduate student, this is many, many years ago that this was created. What does this mean to you as a graduate student at your age of what's sitting behind you?
It's been really cool. I was telling Ryan, this is not something that I've ever worked on. I don't have a background in gastropods or bivalves, but seeing this stuff and seeing the year in which they were discovered and how they were found, fathoms deep or fathoms offshore, it has all that kind of information, and seeing that is really cool. Especially seeing how much of it has been collected and meticulously stored, and the data has just been so well taken care of. To me, it means a lot to be able to see these things that I otherwise wouldn't be able to see. And so it's kind of become a new passion for me, even though it wasn't something I was doing before.
And what do you do?
I work with phytoplankton, so this is very much outside of my realm.
They eat phytoplankton.
They
Some
do
of them
eat phytoplankton, yeah. But it was very different from what I do on a daily basis, and it's become something that I think is incredibly amazing. And I send pictures to my friends. I'll be like, look at this really cool shell, like this is amazing. So I think, and we had grad students come out and work on the annex and get some of the shells out, and all of us had like a field day just seeing these things that we wouldn't have seen otherwise.
So let me also point out that the Sea Lab had a lot of damage from Hurricane Sally. So there was a lot to be done here to recover. So it was not just this shell collection. So we are now, months out, and we're still working on this project. There's still a lot to be done. So I just wanted to kind of add that framework that the Sea Lab did have a lot of damage from Hurricane Sally, and there was a lot to be done on top of all of our our usual work that we do here. So you guys have been tackling this as you're able, but there is some urgency to getting this, to securing this, to getting all of this bagged and matching the shells, with their data, and making sure that we don't lose that to, fading from the sun or to insects or mold. And so, you guys are working as you're able, but we are looking to maybe try to
get some help.
Yeah, some volunteer help with securing this collection.
Absolutely.
So I think Ryan is going to help us with that. Ryan, by the way, was a grad student down here at the lab years ago, and he maintained this collection in his time here. And so Ryan no longer works at the lab, but he is very interested in seeing this collection preserved, and he has volunteered.
It's good to see it in boxes and air conditioning.
His knowledge and expertise and time, to help move this stuff from the cabinets out here, as Randy has been working on, and to get it secured.
So Randy, there's a question for you from Blair. She wants to know what your favorite shell in the collection is so far.
Oh no. I really like a lot of the Murexes that we showed earlier. These spiny guys are really fun. That's just not something, like I said, that I would have seen otherwise, and I think they're kind of cool and dangerous looking. So I like to see those kinds of things.
And Ryan, what's your favorite? You worked with this collection while you were here.
Who's your favorite child? I like the Murexes a lot. They're really, really beautiful. One of the most interesting, we had a bunch of them up here. Let's see, let's take a look. Did we bring out one of the larger ones? Maybe not.
What?
So the cone snails. So these guys, these are...
Some over here.
Did we have... Oh, there's the large ones right here. These are cone snails. Yeah, there's some big ones.
I'm going to set it down where it's supported.
Cone snails, called a cone snail because the profile is a cone. We have some species around here, but they might give you something that is the equivalent of a bee or wasp sting. But some of these specific Pacific species, that Radula, that constantly growing sandpapery like structure they use for feeding, is modified as a little hypodermic needle, little harpoon in these guys. And a lot of these larger species will even prey upon fish. You might have seen these on television. They sit down in the sand and they'll wait for a fish to come by, and they'll pop them with the harpoon that carries a very potent neurotoxin. Actually, it's a very complex venom, and it will subdue their prey very, very quickly, and these things will eat fish, and with other things such as worms and other invertebrates. But we don't have time to go to some of the structural things that are interesting about the shell. I mean, they're very, very heavy and robust, but also again, you're ultimately, despite having a shell, you're a soft-bodied predator tackling much larger flailing prey, and so to have that very powerful toxin helps them take advantage of
people who were unfamiliar with the cone snail's abilities, I suppose, and they, collected them when they found them.
Well, a part of it,
pockets, and then been.
Yeah, so part of it's like if you get a shell and you go, maybe you're on vacation. A week later, you say, "What's that smell?" And there's a hermit crab in it. Well, some of these snails can pull into their shell far enough that you might think it's empty. It might have just been washed up on a beach. There's some stories of people putting them in a pocket or in a mesh bag and carrying them along and getting zapped by them.
And dying.
There are some species that, yes, can be fatal to humans.
So let's go back to that question, Randy. You guys are about digitizing the data. So you guys are working on digitizing this data. However, again, this is a big task, and right now the most critical need is to make sure the shells, and particularly the data, are secured, and then the work can be done to digitize this information.
Yeah,
right?
Absolutely. Like, we have been typing up as we go, but it's nowhere near as in depth as it could be. Like, typing a label is nothing near like typing up those cards would be. So yeah, we're gonna kind of hold off on that until we can save these guys and their data.
So that is maybe an eventual goal, but it's not the most urgent need right now.
Yeah, it would be awesome if we could put it online for people to browse through. That would be amazing.
Yeah, after the rain, it's just making sure it's intact. That's the first step, and then from there, if we can get it digitized. I mean, ideally, yeah, all of these people who have collections like this, to get it online, that would be wonderful. But
it
takes a lot of
way to get it online with that.
You do it by pictures, and you'd have to hand enter all of the information on those cards.
Yeah, because with iNaturalist, one of the things that makes it really easy is that it will put the location data automatically if you allow your phone to, or allow iNaturalist to access your phone. But if you're logging, if you're taking photos of a Pacific, an animal that was collected in the Pacific.
In the 80s, yeah. You have to go back and
input it. It's going to flag that as being collected here.
Yeah. So it's a lot of work.
Do you guys have kind of like a overall message that you think that folks should go away with from the mollusks, from molluscan diversity, from collections, from this particular collection?
Yeah, I mean, I would, as we were saying earlier, I would just encourage people, no matter what you're interested in, whether, again, plants, fungi, insects, shells, just take that extra step and just write a little bit of information down about where you collected it. Because we were talking earlier about, we have boxes of shells, and we say, oh, I'll remember where I collected that. And then you pull something out and go, where was that? So it'll be nice for you, but you never know when a particular species might come up and someone might say, you might read something, hey, this particular species of whatever it is hasn't been seen in a while or is showing up in these places, and to have that data available can be very valuable. And you never know when it's going to be valuable. That's the thing. And so for each person just to take, hey, I got this on summer vacation in 2021. It doesn't take you too long each time you do it. And to have that is really helpful.
Randy?
I would definitely second that. And then I would say for younger folks especially, but really anybody, like if there's something that you don't know about or that you're maybe not interested in, like give it a try anyway. You may find a new passion, because that was me.
Yeah, sometimes you're thrown into things as you were and discover kind of a new path.
Yeah.
So thank you both for chatting with us this morning, and thank you for joining us. And if any of you might be interested in volunteering with us, getting trained, and maybe helping with a collection like this, or helping with something here, then reach out to us through the estuarium, through our docent program. Or you can email me. My email address is mgraeber, that's mgraeber @disl.edu.